listening to the Pharmacy Podcast Network.
Hi, this is Dr. Hector Lopez, and you're listening to the Pharmacy Podcast.
Pharmacy Podcast Nation, welcome back to the Pharmacy Podcast Network, where we're really pushing the envelope here with trying to get a myriad of subject matter experts on so many different discussions. PolitiTalk RX is going to be coming up with a very interesting interview about the world of PBM reform. Everything that's been taking place in the state of Ohio, we're so excited about because we know that when PBM reform actually comes to be, I think we're going to be in a much better situation from a standpoint of lowering drug prices and overviews. But that's not what today is about, because today I'm bringing back a superstar who we had on the Pharmacy Podcast Network on October 30th. October 30th, Mr. Stuart Tomc was on from CV Sciences discussing the science of CBD oil. And Stuart actually has a special guest with him, Dr. Hector Lopez. And I'm going to start with you, Dr. Lopez. Welcome to the Pharmacy Podcast Network.
Todd, thanks for having me on. This is great. Great to be on here.
Stuart, welcome back, buddy.
Hey, Todd. Great to hear your voice.
Great to hear yours as well. We received a tremendous amount of retweets, push, comments, especially from some of the pharmacy students about episode 1 of this 3-part series sponsored by CV Sciences. And your first part, Stuart, discussing the science of CBD oil, really kind of set the stage to demystify this whole CBD oil trend that we're in. Actually, you could probably call it a gold rush of sorts. People really trying to find the best information. Pharmacists out there, primarily our listeners, are being bombarded by lots of questions from their patients. What I'm so excited about and how I'm going to do this today for part 2 is I'm going to turn the show over to you, Stuart. And why I'm excited about that is your last episode just blew me away with the depth, with the direction that we went and the questions that we really shared and came up with. And I was excited about that. So with that, I'd like to hear a little background first on Dr. Lopez. And then after that, the show is all yours, Stuart.
Hey, Dr. Hector, why don't you tell everybody a little bit about your background in the space?
Well, yeah, sure. That'd be great. So I'm a physician scientist by training. I did my medical training and residency and fellowship at Northwestern University in Chicago. After my medical training, I went into full-time clinical practice, founded a few multidisciplinary integrative spine and sports medicine facilities in New Jersey. And then I ended up selling them to one of my partners and actually moving full-time into, I guess, not just my passion, I would say probably, and Stuart, you probably know this more than anyone, probably more my obsession, which is really trying to find a way to connect the dots and bridge the gap between applied nutritional biochemistry on the one hand and really optimizing human health performance in all facets of physical and neurocognitive function, and really trying to improve the outcome of patients when they're recovering or rehabilitating from a clinical condition or healing from a clinical condition. And then for those who are already healthy, my goal is, of course, to elevate the standards of the dietary supplement, functional foods, medical food space in order to help make keep healthy people healthy and make healthy people healthier. So that's kind of my background. I've been fortunate enough to manage and run a CRO, a contract clinical research organization, where we primarily work on validating, substantiating, and executing clinical trials in order to substantiate and validate safety and efficacy of many bioactive compounds multi-ingredient dietary supplements, nutraceuticals, as well as functional foods and beverages there. I'm also fortunate enough to have founded, along with my partner, I co-founded Supplement Safety Solutions, where we do nutravigilance. And nutravigilance, I'm sure for anyone in the audience who understands what pharmacovigilance is, nutravigilance is the equivalent in the dietary supplement nutra space instead of pharma. So we do post-market safety surveillance, and we've got a ton of data that we've been able to amass over the last few years, and just very fortunate there. Also, the co-founder of THR Biosciences, where we do novel ingredient discovery, incubation, and development in order to bring evidence-based novel ingredients or ingredient matrices to the market and license them to the market. So that triad is really where most of my professional time is spent these days.
Todd, can you see why I asked Dr. Hector to join me on the show today?
Yeah, that's awesome.
Let me give the historical background for the storytelling, because it's a big part of it. I had the great privilege to meet Dr. Hector Lopez when I was employed at Nordic Naturals. He was a member of the medical and scientific advisory team. And so I had good fortune to work with him then. And I was really struck with this. It's more than an obsession. I mean, it's a call, a deep life calling, Todd, to not say stupid stuff, but I spell stuff a little more poetically. How's that? Okay.
Good.
So here, I am very hyperbolic. I'm very emotional. I'm very, very excitable, which makes me good at what I do. However, when I ran into Dr. Hector and I was the spokesperson for Nordic Naturals, I ran into a human being who basically demonstrated to me, modeled for me, what it may look like, what I may look like, what we may look like if we just stick to the science. If we don't exaggerate the science in any way, shape, or form, just get excited about the science. He was the person that modeled that for me. So he has become our key opinion leader here at CV Sciences. All of the toxicology work that we talked about last time, the formal investigation to demonstrate the safety of the ingredient. Dr. Hector has been at the forefront of all of that work as the coordinator between the research groups that we hire and the different stakeholders. Dr. Hector has really been the leading vision and scientific voice that has really led CV Sciences to where it is today. So that's why I thought after we were able to get people to come back, this time we would have a far more specific, more pharmacy-oriented conversation around the red, green, and gold label, and also to help people better understand the difference between isolated CBD, which is very prevalent in the market today, and these food-based hemp extracts that happen to contain naturally occurring cannabinoids. How are they used and how are they different from isolated CBD preparations? So I thought we would bring the smartest person that we knew on the subject to your podcast to help break it down, deep dive this time.
Stuart, that's incredible. Very much appreciate that. And I know my listeners are going to really appreciate that. And the reason being is collaborative initiatives between pharmacists and physicians are bigger than ever. And we know that patients are constantly asking pharmacists about the world of CBD oil. So you guys take it away. I'm going to sit back and get my popcorn and sit here and listen.
Well, the first thing that I want Dr. Hector to address is the difference between isolated, pure CBD, which is widely available now as a pharmaceutical prescription and also in health food stores. I just want Dr. Hector to speak for 5 minutes uninterrupted to really get to the kernel. What is the difference between 100% isolated cannabidiol CBD and these hemp extracts that we are making? Now, we're not going to have the time today to compare all of the different offerings in the marketplace because there are many of them. We're going to go with the general principle that there are 3 different swim lanes. The 3 different swim lanes where hemp and CBD will be sold. You will find pharmacy products where CBD will be sold. There will be medical marijuana dispensaries. That's not what we're here to talk about today. But those places have high CBD, low THC marijuana products for sale. And those are different from the hemp extracts that CV Sciences is bringing to the market. And with the imminent passing of the Farm Bill, it's very important that you know the difference between a hemp extract, considering hemp as an agricultural commodity is about to be completely removed from the Controlled Substance Act forever, that you know the difference between what a hemp extract is, a marijuana extract, and an isolated CBD product. So in order to get there, we're going to start with just the isolated CBD molecule. So Dr. Hector, if someone was just to ask you, a patient or someone just walking down the street, hey doc, can you please tell me the difference between isolated CBD and these hemp extracts by CV Sciences, for example, the Plus CBD Oil Gold formula? If you could compare our gold formula extract to isolated CBD, please.
Yeah, no, that's it's a great point, Stuart, because we really do run into that often. There's a lot of confusion currently in the marketplace, and to be honest, I as you know, Stuart, I just got back from a medical conference where we presented, and I was part of the symposium on cannabis and medicine, and we really reviewed extensively the entire endocannabinoid system, and then we talked about the various products that are available, including isolated CBD, the molecule CBD isolate, versus these. I've kind of changed my vernacular a little bit even. I've evolved from calling them full spectrum hemp extracts to just being a little more careful, and I think I think we're probably better off calling them broad spectrum hemp extracts, because just by definition, what does full really mean? Have you characterized all 116 plus phytocannabinoids in the product, or is it just a broader hemp extract with many more bioactive constituents?
You're right. That's more precise language than full plant.
Right. Yeah. So I've kind of evolved to using that term, I think, a little bit more recently. But look, I would probably start by telling a patient that you can think of, and these are just analogies, right? So there are many things that are inaccurate about an analogy, but I think just that if we want to talk about an analogy, I would consider isolated CBD like an active constituent, an active ingredient, an active compound, like an alkaloid, for example, in coffee. So you've got caffeine, right, anhydrous caffeine. Trimethylxanthine, right, is caffeine, 139, I believe. 137, I'm sorry, trimethylxanthine is caffeine. That's the chemical compound known as caffeine. But within coffee, you have a cornucopia of many other compounds other than just caffeine. It's really essentially a witch's brew, if you will, right, of many other alkaloids. There are many other polyphenols. There are many other compounds and volatile compounds, as well as non-volatile compounds within coffee, a cup of coffee. So that is akin to the isolated CBD product versus in its most raw form, a hemp plant raw crude extract, that first stage extract, for example, like what would be what you would see in the red label CV Sciences Plus CBD oil product.
Wait a second, doctor, just only because this is so complicated. We just want to try to compare. If a pharmacist is looking at, for now, we're going to get there, our gold label soft gel, 15 milligram soft gel to isolated CBD, just so that when they're looking at that, just so I can give some more context, the pharmacist picks up our number one seller, our 15 milligram gold soft gel, and they look at the 15 milligrams per serving, and then they read the New England Journal of Medicine that says the amount of cannabidiol that was used for treatment-resistant seizures was upwards to 20 to 50 milligrams per kilogram.
Right.
And so that then the confusion rockets just go through the air. So when you're looking at our gold soft gel versus isolate, let's just stay on that track just for a moment.
Sure. Yeah, it's a completely different product, right? Because the gold isolate is essentially a material that is 25% CBD by weight. So you still have 75% of the rest of the product contains other constituents, right? Now, obviously, there are some fatty acids in there. There's some very long chain and interesting fatty acids, almost wax-like fatty acids in there. But there are also all the vitamin E isomers are found in that gold product that you mentioned. There are also some flavonoids that are found in that gold product. There are also other phytocannabinoids besides, or in addition to CBD or in addition to cannabidiol that are found in that gold broad spectrum product. So you have a broad spectrum of compounds. There are even some terpenes also. There's some minor terpenes in there as well. So you have this kind of array of other compounds that are found in your gold product that you do not find in a 99.7+% CBD isolate products such as Epidiolex or the compound, the drug that has recently received FDA approval for drug-resistant seizure and recalcitrant seizure and Dravet syndrome, etc. So that, I think, that is a very important point to realize, that when you look at the back of a bottle and you see 15 milligrams, but it's 15 milligrams of CBD in addition to possibly, would you say it would be maybe 5 to 7 milligrams of other phytocannabinoids possibly, maybe a little less. But there may also be many other milligrams per serving of terpenes and the vitamin E isomers and some other flavonoids as well in that one dose, one serving. And the reason that's important is really it goes back to that paper, Stuart, I'm sure you know very well, you're familiar with, the Galilee paper with the whole plant conversation that we often talk about. So
can you talk a little bit more about that as we're strengthening this argument? I know we're getting closer to it. Can you talk a little bit about that paper and that where they compared isolated CBD to full or broad spectrum extract that contained natural CBD? What observations did they make? That was remarkable, wasn't it?
It was, actually, because what it showed was that normally, if you take isolated CBD, you basically get this non-linear, and of course for pharmacists, they're very familiar with dose-response curves, right? And pharmacodynamics. And so unfortunately, you get this non-linear, almost bell-shaped response, dose response, when you apply isolated CBD to some sort of a disease model. Let's use the disease model that, for example, they used in this Galilee paper was they basically caused the inflammation to the paw. So they used the writhing response of an injured or inflamed paw of a rodent model. And so what this study showed was that in stark contrast to purified CBD, where purified CBD has a bell-shaped non-linear curve, in terms of the more ingredient, the more CBD you give, you don't necessarily get a linear response on a reduction of pain and inflammation and nociception or analgesia. What was found is that a whole plant extract that contained only a fraction of the CBD compared to the purified CBD, when it was given orally even, provided a clear, linear correlation between the anti-inflammatory and anti-nociceptive response and the dose. Now, that's important for any clinician, right, who's treating patients, or even a non-clinician who just wants to be able to know that you can have a reliable linear increase in your dose, in your response, when you increase your dose. And I think that's one of the strongest arguments for the evidence that there is the existence of this entourage effect that Dr. Raphael Mechoulam has talked about for decades now. Right? So,
before we define that entourage effect, the synergy of these things working together, let me just summarize this before we move on to make sure I understand this. Are you saying that isolated CBD, we're probably going to see a bell-shaped curve in terms of dose response, versus these broad-spectrum plant extracts, hemp extracts, that contain all of these cofactors seem to have a very elegant sort of linear dose response? Do I understand that correctly?
That is exactly it. Yes. Yep.
Okay, so would that then stand to reason, since we're speaking just to pharmacists, that if a patient came in that was dealing with treatment-resistant epilepsy, our very first recommendation would be to get a prescription for the newly approved Epidiolex, which is 99% CBD and 0.1% THC, because that product is designed to be taken with other drugs, and you get direct insurance reimbursement, you hope, child-proof cap, and clear directions. That would be different than somebody who, say, was looking for a natural way to deal with general anxiety disorder, dealing with systemic inflammation that doesn't seem to resolve itself, that they're just completely different patients with completely different needs, and that part of the confusion is it's all called hemp extract. Do I understand this correctly?
That is well put, and absolutely, that is correct. You certainly understand the scenario and the argument. So it really is a different product with a different purpose for a different population and a different indication.
And that is so critical, because we've never had, or I can't remember, a chance to navigate something as complicated and as nuanced as this. Because think about what we're saying. We're saying, here, see this dose used in the New England Journal of Medicine? That's not the dose you need. You need far less, you see, because over here, this natural product contains all of these cofactors. Now, it sounds like that's magic, right? That doesn't line up with the deep rigor that we promised everybody at the top of the show. So how do you square that missing information in the natural hemp extract conversation versus the certainty that we demand with evidence-based medicine? How do you reconcile that?
Well, for one, I think this Galilee paper shows us that the existence of quote unquote the synergy that may be occurring when you combine the host of other phytocannabinoids in the presence of cannabidiol or in the presence of CBD is there. But in terms of how do you reconcile it mechanistically? Most pharmacists are aware of structure-activity relationships. They're aware that receptors and enzymes have allosteric sites on them. And they also understand, I think, that in a sophisticated fashion, if you have a multi-ingredient product versus a single-ingredient product, it's much easier to drill down on a single compound, a chemical entity, a single chemical entity, with a single target with its own structure-activity relationship. That's very different than a multi-ingredient, complex, multi-constituent product, which we're talking about when we refer to these broader hemp extracts that contain all these other constituents. Those other constituents are doing things. They're not inert, right? We're not talking about compounds that are excipients. We're talking about multiple bioactives in one product. And the fascinating thing here is that you would anticipate that when you have multiple bioactives in a product, that it might create a quote-unquote dirty drug, which many research pharmacists are also familiar with from a vernacular terminology standpoint. But in fact, what the data and what the evidence is showing us, the available data, is actually showing us that the cleaner response, the cleaner pharmacodynamic response, is being observed when you use a broader plant extract, a broader spectrum of constituents. You're actually getting a more reliable linear response when you increase your dose, as opposed to using the isolated one molecule, one compound. And
so if we're trying to make an argument that you're going to have a cleaner, interestingly enough, considering the term dirty drug,
right.
a cleaner response with the difficult to measure signal to noise ratio complex biochemical matrix. Let's now get into our thought machine. Let's expand our brains and let's use an analogy I think will help to really make your point, Doctor. And let's talk a little bit about the recent work that Ethan Rousseau did, where he was looking to make your point, to compare synthetic THC to full plant natural CBD and THC with other cannabinoids in the Sativex pharmaceutical product. Sativex is essentially cannabis in a spray bottle. And the way that we compared full plant natural cannabis extract to isolated synthetic Marinol, considering that's a drug that every pharmacist listening to this should know. Why don't you talk about the observations made in that study? I think that really helps to make your point.
Yeah. So the study that comes to mind is probably the one where they use the nabiximols or Sativex, the THC CBD more broader plant extract versus THC in isolation in intractable cancer-related pain. Is that correct?
Yes, where they showed the toxic dosing psychosis threshold.
That's another one. That's another one. So the one
do both if you want.
Right. So there's one that I'm thinking of, which was a multi-center, double-blind, kind of gold standard placebo-controlled parallel group study, looking at the more full extract, cannabis extract, which included a 1-to-1 ratio of THC to CBD, versus THC in isolation, as an isolate, in patients with cancer-related pain. And it showed that the response was substantially improved in the combination of THC plus CBD plus all the other phytocannabinoids and other terpenes that were included in that extract, which I think often gets lost in conversation because they are in fact present in that Sativex nabiximol product versus THC alone. There was over a 20% increase in synergistic response to pain in the intractable cancer-related pain patient population. And that was using the same total milligram amount of THC and cannabinoid in one product versus the other. So clearly there's that showed that there was a pharmacodynamic synergy. And then the other study actually showed that there was a much higher margin of safety. So the therapeutic window, the therapeutic index, was actually much higher in the combination THC plus CBD plus terpenes and all the other constituents product versus THC alone. So that the presence of CBD and some of the other phytocannabinoids plus some of the other terpenes, what it did was it actually helped to mitigate some of the psychotoxic effects of THC when it is utilized in isolation or at too high of a dose.
So just so I understand correctly, when I'm looking at this and it shows Marinol, something again we're all familiar with, at about 15 milligrams, patients started to report this toxic psychosis threshold. So it had a real limited safe dosing range. However, when we compared that isolated THC in the Marinol to full plant CBD THC extract in the Sativex, they were able to get almost to 50 milligrams without hitting that toxic psychosis threshold. So do I understand that the CBD and the other cannabinoids and cofactors and terpenes actually modulate and buffer some of the effects of THC so that we can really maximize its effect when it's working in concert together as it appears in nature more effectively than, say, isolating the one molecule by itself? A, and B, is that the reason why pharmaceutical companies stopped exploring these cannabinoids for drugs years ago? Is it because they just couldn't make them work through their paradigm?
Yeah, those are actually all great points, Stu. So yeah, to your first point, that is accurate. So synthetic THC in isolation had a toxic psychosis threshold of about 15 to 16 milligrams or so. And then the combination product that had more of the, let's call it the broader spectrum cannabis extract product, the nabiximols, that had a toxic psychosis threshold of nearly, not quite, but nearly 50 milligrams. So you're talking about, over a threefold improvement or broadening of the therapeutic index or the margin of safety, just because you included the other constituents of the plant. Now, in terms of mechanistically why that may be happening, we actually have a good working hypothesis, and there's actually data supporting this. And the reason for it is because the presence of cannabidiol itself is actually an allosteric modulator to the CB1 receptor. So that means that when you have THC in isolation, THC is an orthosteric agonist to the CB1 receptor. It fits very well and very effectively into the main binding pocket of the receptor, right, of the G protein coupled CB1 receptor. However, CBD or cannabidiol actually interacts with a different site, an allosteric site, which is outside of that main pocket, that main orthosteric site, and it slightly changes the conformation or the shape of that receptor. And by changing the shape of that receptor, what it does is it negatively modulates the signal downstream of that receptor. So once you flip that switch and you've turned that receptor on, it's almost like having a dimmer on the switch when CBD is present. Does that make sense?
Doc, is that why anecdotally, people report that when they've taken too much THC, they take a lot of CBD or our gold formula hemp extract, and they claim that it makes them sober again or normalizes them?
This certainly would provide a very good rationale for why that might be, why that is something that people are observing anecdotally. But yeah, this would provide a reason for it. Absolutely. It's like putting a dimmer on a light switch that otherwise would simply be an on and off. And when it's on, boy, it can be intense, and that downstream signaling could be intense. So, yeah.
So are we to take from this part of this that the cleaner option, if you will, in terms of the well-being of the patient may actually be these broad spectrum crude artisanal hemp extracts, unless there's a very, very specific reason that we would want to write a prescription for, say, Marinol or Epidiolex? Different product, different customer, different market, and we shouldn't confuse the medical patient getting a prescription filled at the pharmacy with somebody looking to use hemp extracts to deal with anxiety, stress, and inflammation, correct?
That is correct. Yeah, no, that's a very important point. It's not to say that there isn't a role. I see them as, they're different tools that you're going to pull out of your therapeutic armamentarium or out of your toolbox. And you're going to use those different tools for different purposes, right? If all you have is a hammer, all right, then everything around you looks like a nail. But in this case, you've got a hammer, you've got a screwdriver, you've got a little bit of a chisel, and you really can get creative with using a different product for a different indication and a different purpose and a different patient, certainly, or individual who's presenting differently. But
okay, so I'm going to hit you up here with a couple of questions to see what you would recommend or what you think someone could consider. But before we do that, I want to help recalibrate all of our brains. We're talking about the difference between isolated cannabinoids and these broad spectrum plant extracts, and we're comparing isolated CBD to our 15 milligram gold soft gel, our plus CBD oil soft gel, which is our number one selling product. It contains 60 milligrams of hemp oil that contains 15 milligrams of CBD, and the capsule also contains a rich, abundant source of fatty acids, terpenes. The dominant terpene in the hemp extract is beta caryophyllene, one that is found in black pepper and tastes like wood. We also have all 8 isomers of naturally occurring vitamin E, and 50% of the product is fat and fatty acids at this point, also increasing bioavailability. We mixed it with extra virgin olive oil because that also helps to support the endocannabinoid system directly and indirectly. And also there was a qualified health claim that came out. I know you caught that, Dr. Hector, that extra virgin olive oil may reduce the risk of coronary vascular disease. So we're lucky we chose extra virgin olive oil instead of other delivery systems in our product. But that 15 milligram, say it again, Doc?
No, I was going to say good choice. Absolutely.
That worked out. That was perfect. But our 15 milligram gold soft gel, you've got a pharmacist that just got their hands on the New England Journal of Medicine's paper. They didn't have time to listen to this podcast because they're busy. They're looking at the New England Journal of Medicine paper, and it says 20 milligrams per kilogram a day of body weight. 20 milligrams per kilogram of body weight per day for CBD. And you hold up a 15 milligram gold soft gel, and she's very confused. She has no idea, why are you recommending such a small amount of CBD? Why would someone want the high dose CBD, and why would someone want the gold soft gel?
Yeah, so obviously the high dose isolated CBD has reliably been studied to reduce the incidence and the severity and the magnitude of treatment-resistant or recalcitrant epileptiform activity, or epilepsy, right? Drug-resistant epilepsy, conditions such as Lennox-Gastaut syndrome and Dravet syndrome. These are conditions in which the brain is it's hyperexcitable. There's a lot of excitatory neurotransmission occurring across the hippocampal region of the brain, and hence it sets off a very serious condition of high frequency intense seizure activity. However, the other product, the 15 milligram gold CBD dosed product, you need to think of it as it's not just CBD. It's CBD plus many other, let's call them cofactors, many other phytocannabinoids. There are other fatty acids. There are vitamin E isomers, as you say, and they're all 8. There's also the presence of other terpenes, the predominant one of which, of course, is beta caryophyllene. And the presence of all those other constituents make this product more suited for immune modulation, for improving the discomfort associated with physical activity or intense exercise, for example. It may improve joint health. It may have musculoskeletal medicine benefits. It may even have metabolic health benefits based on a lot of the preclinical research and even some emerging clinical research that we're starting to see. It can have some stress resilience or a healthy stress response benefits as well. So I think that's probably a good way to distinguish or delineate a broader spectrum hemp extract product like the gold CBD plus CBD oil product with 15 milligrams versus a CBD isolate 99.7+% CBD isolate product. at a much higher dose, of course.
So would you also then talk about some of the short-term versus the long-term benefits? Because I think that there's a little misunderstanding here because of our lack of familiarity with the endocannabinoid system and all of this crosstalk. But do you see there being some really untapped benefits for long-term use, which we've not yet been able to capture because this is so new, versus what you're seeing in terms of being able to help people in a pinch with, say, stress?
Oh, yes, absolutely. So I think the best way to describe that is the fact that the endocannabinoid system itself, the signaling, what happens downstream of signaling cascades in the endocannabinoid system, it occurs in multiple waves. So usually the first wave of which may happen is if you have an andamide is more of the sort of stress responsive endocannabinoid, and 2-AG is kind of the workhorse endocannabinoid of the nervous system. So oftentimes if you need some acute relief, then the presence of something like the CBD in the gold oil, or even the green product, the green total plant complex product, they may benefit the acute signaling cascade and allow for sort of a stress response or anxiety benefit. Then long term, there's a second wave and third wave that occurs, which is what happens after the downstream of the receptor, downstream of the cannabinoid CB2, CB1 receptor has signaled. There are gene expression changes that can take place. And there are multiple genes that can be upregulated and others that are downregulated, which may have a longer term response with respect to inflammatory modulation or supporting a healthy immune response or a healthy inflammatory response. So does that sort of answer the question about some acute effects versus some longer term benefits? There's also the way that the body handles nutrients as well. So nutrient partitioning, we like to call that, the way that the body metabolizes carbohydrates and lipids as well. That can be affected long term by a healthy endocannabinoid tone.
Thank you. No, that was very important because the gold is our number one seller, and it is so fast acting and so effective. I think, unfortunately, a lot of people stop there and they think everything is treatment resistant. And we're finding out with our experience now doing this for years. You mentioned the Nutravigilance. Dr. Hector and his team record any adverse events. So with selling our products, if there's ever any adverse event or side effect of any kind, please call the 1-800 number on the side of the package so that we can record all of that information. That is really important information. And one of the things that we're hearing, Dr. Hector, because this product is popular and people are in pain, often they miss the benefit of fine-tuning the endocannabinoid system by taking 3 gold soft gels immediately and missing the benefit of low and slow. So I wanted to start with the treatment-resistant gold product because it's our number one seller. If you're dealing with treatment-resistant conditions with mood and psychogenic components, migraine, irritable bowel, fibromyalgia, stuff that won't budge, the gold is where you should start. But that is not for everybody because we're working in the dark and we can't tell where your endocannabinoid system is activated, how dense or thin the receptors are. So isn't that the reason why ultimately, if people have time, they can get even better or improved results by starting very, very slow instead of going right for the gold soft gel? So that's what I wanted you to talk about as we bring it home, about using either the red product and why that is different, and the green product, and why some people may only need a very, very small amount to get the benefits that we're looking for.
Yeah, no, I think those are important points to cover. So first, I'll lead with the fact that you can get an enormous amount of variability, right, in terms of the therapeutic margin or window. The therapeutic index is very broad, which is good from an adverse event standpoint and from a safety standpoint, right? Whenever you have a drug, or in this case, a natural product that has a very broad therapeutic index, then what that does for you is it gives you a very strong sense of confidence and certainty that that even if you use a dose that is beyond the least amount necessary, that you're not going to push someone towards a severe, or certainly a serious adverse event. So that's one point I think that's important to make. The other is that while some patients may respond dramatically to as little as, I think I've told you these stories in our research center, you have some patients who respond to as little as 1.5 to 3 milligrams of active CBD from either a red label product or a green label product in the oral mucosal spray, for example. And then you have that same type of condition in a different individual who may not respond to 15, but may need 15 to 30 and may need to push the dose and titrate the dose up a little higher. And so you do get this variability in dose response, but it's inter-individual. It's from individual to individual, or from patient to patient, from consumer to consumer. And then finally, I'll say that starting low and going slow is certainly the sine qua non of how you manage anyone who's starting to utilize cannabis extracts for treating or modulating some sort of endocannabinoid imbalance or a clinical endocannabinoid deficiency, right? As Dr. Ethan Rousseau has published on. So I would say starting low and going slow, there is value to understanding that even small doses can be incredibly effective at modulating inflammation, mood disorder, stress response, and stress resilience. So you can start having subjects or patients responding to as little as 1.5 to 3 milligrams, and they can be titrated all the way up to being comfortable at 30 to 45 plus milligrams daily. I will say something about the red label, though. The red label product is unique in that it actually is mostly standardized for cannabidiolic acid, or CBDA, as opposed to the decarboxylated CBD. And CBDA, that carboxylic acid group off of the parent molecule of CBD, that in and of itself actually causes some unique or different mechanistic sort of characteristics for the ingredient. It actually tends to first stay more in the periphery. It's less likely to pass the blood-brain barrier with the presence of that additional carboxylic acid group. And moreover, it tends to be a more potent modulator of the COX and LOX enzymes. And so, of course, many pharmacists are very familiar with cyclooxygenase and lipoxygenase enzymes, and CBDA tends to be a more potent modulator of that enzyme. It also tends to be actually, and I've actually observed this empirically again in patients, is it tends to be a more potent, it has a more potent antiemetic effects as well. So it tends to decrease nausea and perceived nausea, as well as active nausea and vomiting in many consumers. So I think those are interesting notes about the red label, and the opportunity to use both the red label in conjunction with the gold label, for example, in certain customers as well, because they have slightly different characteristics, but they can be complementary of each other.
Well, thank you very much. I also wanted to mention Dr. John Hicks. his work. He's an MD who's no longer with us, but he left a great book, The Medicinal Power of Cannabis by John Hicks, MD. And he used the green spray, the 100 milligram spray, peppermint flavored, one spray, to your point, sometimes two sprays with children. He was a pediatrician seeing kids all over the world, and he's the one that used to say less is more. Less is more. So thank you for pointing that out, and then also the wide range of dosing. And we want to add to that the really exciting paper that came out earlier this year in the European Journal of Drug Metabolism, Pharmacogenetics of Cannabinoids. So make sure that you understand, we all do, the gene variations. Dr. Hector talked about wild differences between us. Gene variations are responsible for the presence of the therapeutic and side effects of medical marijuana and cannabinoid-based drugs. So look forward to some 23andMe additions on the cannabis cannabinoid SNPs to help better understand how this stuff is operating in us. Doctor, do you think that that's a far stretch, or do you think that we'll end up having some of those, transporting SNPs and those other things? We'll be able to look at those through tests like 23andMe in the not so distant future?
Oh, absolutely. I think that is certainly coming. I think it's going to be around the corner sooner than later. And as a matter of fact, I think we'll probably even be seeing maybe a metabolomics-based biomarker analysis soon of the endocannabinoid system, right, in terms of measuring biomarkers of the endocannabinoid system in plasma and blood. The way that we almost do for omega-3 blood spot testing to look at omega-3 to 6 ratio and fatty acid status, omega-3 and omega-6 fatty acid status. I think we're heading in that direction. But yeah, anytime you have a system like the endocannabinoid system that is based on the presence of key receptors, regulatory enzymes, then you obviously have a strong epigenetic component as well as pharmacogenetic component there to consider. So that's coming. That's definitely going to be coming.
So until then, and even then, the onus is on you to figure it out.
Right. Yeah, that's why starting low and going slow is going to be the best advice.
And there are a few little hacks that we want to share with you here to improve your health if you're trying to work with your endocannabinoid system and you're not getting the results that you want. In addition to aerobic activity, which not everybody can do, but has been noted to increase endocannabinoid tone. Probiotics seem to have a very positive effect for endocannabinoid tone. Highly unsaturated fatty acids, if they're the right kinds. We certainly know that the backbone for the endocannabinoids are arachidonic acid, and there's even been synaptomide has been identified from DHA, and there's some EPA derivatives as well. So long-chain EPA and DHA may also be helpful. Something like turkey tail, also to help feed some of the microbiota and help deal with some of the immune signaling. Those can all be nice tools to help improve your endocannabinoid system, and as a result, your CBD and hemp extract results. So remember, it's also about nourishing our body in a new way. Hemp is not new. The endocannabinoid system is not new. I mean, it's relatively new, 1992, but it wasn't discovered yesterday. But people have been neglecting their endocannabinoid systems, and they're not feeding it or taking care of it, are they, doctor?
No, absolutely. I think that's an important point. Is the system has obviously been there since the very first organism that displayed a primitive nervous system, right? The sea squirt and lamprey. The endocannabinoid system has been present. It's there to respond and react to challenging environmental dynamics, and certainly, physical activity has a major impact on your overall endocannabinoid system health, as does your background diet. Again, the omega-3, omega-6 functional fatty acids you mentioned are critical. Gut health, and just your general overall inflammatory status, right? So somebody who's got metabolic syndrome and has some baseline systemic inflammation is going to have probably some more perturbations in their endocannabinoid system than other high-functioning individuals who follow a very wholesome diet and are very active and are highly functional and aren't walking around in chronic pain or systemic inflammation. So,
at the risk of oversimplification, if you're healthy, your endocannabinoid system is healthy. If your endocannabinoid system is healthy, you maintain a stable weight, a stable mood, and have regular bowel movements, you might not need cannabinoids. You take cannabinoids, they might make you feel weird because your ECS is already balanced. Is that too much of an oversimplification?
No, I think that's fantastic, actually. I like that. That's a great take-home message.
So let's remember that this is about nourishing the ECS to help you feel better. And sometimes CBD can help. For healthy people, start small and work your way up. Think adaptogenic, homeostatic, supportive, nourishing. For those who need more, for those in need of advanced need, well, then take more. I mean, feeding your body supplements and foods that boost the endocannabinoid system might mean that you need to take less CBD. That is our 2019 message. Feeding your body and supplementing the foods that it needs to boost your endocannabinoid system might mean that you need less of the product every single day. So nourish your body with a broad spectrum hemp extract. And thank you for letting us do such a deep dive today, Todd, on the Pharmacy Podcast.
Yes, Dr. Lopez and Stuart, you guys should have your own show. This has been absolutely amazing, especially with the Congress just passing the 2018 Farm Bill. There's less and less stigma. There's more and more data. And I see things coming to light, especially with the leadership that pharmacy can place in between the physician and the patient to supplement and enhance that relationship as well as the education. So I want to thank you, Dr. Lopez, for being a guest with Stuart on the Pharmacy Podcast and really blowing out the information side of this. Stuart, I'm going to want you guys to come back. Obviously, part 3 is coming up. But wow, I just think you guys did an amazing job.
Well, thank you. Thank you so much for the kind words there, Todd. It was fun. It's always fun when we get a chance to take some of these deep dives. And obviously, Stuart, is great at being able to pull me out from the weeds sometimes if I get in a little too deep. So,
well, I once again want to thank Dr. Lopez and Stuart Tomc from CV Sciences sponsoring this 3-part series. Stuart, we'll hear from you again very soon.
Thank you, Todd. This was one of the best conversations we've ever had, and it's recorded, so please share this with your friends.
We will. And with that conclusion of today's podcast, please tweet us at Pharmacy Podcast just to ask any questions, and there will be show notes so that you can reach back out to Stuart Tomc, part of the CV Sciences team. And thank you all for listening to the Pharmacy Podcast.